Good morning. Have a nice day. I am Nemerix Gerson. I will be presenting improving photosynthesis and metabolic networks for the competitive production of phototroph-derived fuels. So this is a research, a paper published by the Environmental Science and Engineering Division, Colorado Coal Mines, Illinois, Street, Colorado, USA. So the authors are Victoria Work, Sara D. Adamo, Randor Randokovich, Robert E. Jenkerson, Matthew C. Uh, Puswitz. So this is also published in the Elsevier and in also in ResearchGate. So you could access the whole paper in the given link. So for the introduction, for the interest of the use of PSMS or the photosynthetic microorganisms as feedstocks for bioenergy, which the bioenergy comprises the biodiesel, the biogas, or the biofuel, and the bioalcohol, has been emphasized and demonstrated repeatedly, especially within the past 15 years. So, their manifold attributes include high phototropic productivity, diversity of environments conducive to growth and production of simple biomolecules, easily converted the fungible fuels into its manipulation in order to have more starch to process, and the processing of this non-cropland species in a biofuel rather than using plant biomass as its source. However, an industrially competitive system has yet to be established. An aggressive An aggressive approach is needed to develop the strengths of suit mechanisms of the CCM, or this is the central carbon metabolism into C3 plants. Though true C4 metabolism relies on the differentiated cell types, this Thalassosera wisflogi is a C4-like system, which is a diatom, and this is detected. No? Simpler CCMs has been detected in many microalgae like the Thalassosera. So this species, this microalgae, is a species of centric diatoms, and it's found in the marine environments, also in inland waters in many parts of the world, such as they are also cultivating this, no, to produce these micro algae, because it's C4 plant style strategies in increasing its photosynthetic efficiency. So as you can see in its physicochemical parameters, there's a high volume or amount of alcohol soluble extracts, which is 11.4 grams in 100 grams. So here are some image wherein the algae can be increased its photosynthetic activity by cultivation. So as you can see, they're in a big basin exposed to sunlight in order to increase its photosynthetic activity rather than obtaining those microalgae in the in its natural habitat, like the inland waters and the sea waters. So fundamentally, careful design introduction of high activity versions of CAs, or the crassulations acids, that is a product from the CAM photosynthesis, this C fixation to adapt the arid conditions, are also found in some microalgae, no? and as well as chloroplastic bicarbonate transporter could greatly augment carbon dioxide concentration around the ribosco. And we know this ribosco is the enzyme that is incorporated in the carbon dioxide into plants' photosynthesis. This directly enhances the efficiency of energy transfer from photosynthesis to carbon fixation and its improvement of ribosco's specificity and turnover rate that would promote this effect in order to increase the photosynthetic activity. So the photosynthetic efficiency is required for the production of all potential bioenergy surrogates from PSMs, or this is the plant specialized metabolites, no? So this plant specialized metabolites are involved in various physiological and biochemical processes, no, to adapt and defend the adverse environment, no. So it is relatively low efficiency process in terms of energy conversion when compared to downstream synthesis of targeted products. So why are they cultivating this algae as a source of a bioenergy? Well, it would be an alternative source from those corn and grain biomass, no? So more than 90% of photon energy delivered to given photosynthetic footprint can be dissipated as heat fluorescence, and current estimates for realistic photosynthetic conversion efficiency fall around 6% of the total light incident. Thus, the more photon energy delivered to given photosynthetic footprint means more glucose to be developed. We will check this example. So as you can see in the image, more photosynthetic activity is found higher in a high light conditions, no? And it was found out that algae has higher photosynthetic efficiency than the higher form of plants, no? Because of their antenna pigments that would harvest the solar energy. That's why research institutions nowadays are cultivating the some microalgae as a source of biofuel. Okay, so the lipids. So the lipids or the TAG as an attractive biofuel feedstock also is a major storage lipid produced by the microalgae. Much effort has gone into investigating lipids photosynthetic pathways in algae, and several attempts have been increased their lipid productivity, such as the starch biosynthesis in the Reinhardt tea. No? This we know that the starch is a chain of glucose, no? So that's the thing that we want to extract to produce a biofuel from the biomass of the algae. So like this Chlamydomonas species are widely distributed in the soil and fresh water. So this Reinhardt tea is a species especially well studied biological model organism due to its is culture, no? It is easily cultivated or cultured. So the interest also of producing biopharmaceuticals and biofuels, as well as valuable research tool in making hydrogen from this species. So for the carbohydrates also, it is a primary store of photosynthesis in many species of algae, serve structural and transient energy rules, no? like the starch. Okay, so the carbohydrates such as cornstarch have many industrial applications, have long been a feedstock for biofuels through conversion to alcohols, no? Through fermentation or hydrolysis. So here are some algae which is enriched predominantly as a storage of polysaccharide, like the green algae or the chlorophyta, the dinophyta or the dinophytes, the glaucophytes or the red algae or the rhodophytes. So the generic chloroplast of green algae showing placement of fuel relevant primarily the metabolites and their integration of bioenergy production. As you can see in the image, extracting the starch, no, to produce alcohols, biohydrogen, biodiesel, and the biodiesel additives from the isopropanes and from the TAG as we mentioned a while ago. So in the perspectives of using or developing a high photosynthetic activity in algae, well, some biofuel research major advances are being made in pathway engineering for desired products, no? So while it is a focal point, the task of optimizing the photosynthetic efficiency of these PSMs has not been aggressively pursued, and it is only starting since 2012. So this could be due to fact of the conversion efficiencies of algae already higher than land plants, or it may be stymied by the complexity of the manipulating the photosynthetic apparatus from the algae, from developing algae as a source of biomass for biofuel. Okay, so before I'll end the lecture, in the end process, we have come up with fuels, algae-based fuels, isobutanol-based fuels, and other fuels that we think will power the planes in the future, so that, you know, by 2020, I hope that our planes will be powered on the fuels that are clean and not polluting the environment. So the green airline and an airline that actually has fuels that will be hopefully cheaper than the dirty fuels of the past or the fossil, no? And so we're doing good and also turning a profit at the same time. By Richard Branson. Thank you very much for listening.